Thank you for the introduction. It's a pleasure to be able to talk a little about the REPHRAME project, which deals with one of the major pests of pine forests in Europe and indeed around the world. As you can see, the title of the full project is Development of Improved Methods for Detection, Control and Eradication of Pinewood Nematode in Support of EU Plant Health Policy. And that's one of the key aspects that we will be dealing with, is that the information we've gathered in this project is being used by the European Union to develop plant health policy. The project itself involves 11 beneficiaries, as they're called. These are the partners in the project from 8 different countries, and here we've got a list of them all. A couple from the UK, a couple from Portugal, a couple from Spain, and then several other countries. The title I've already mentioned. In fact, this problem has been going for some time, as I'll detail in a moment. And we had a previous project called FRAME, so this one is called REPHRAME. And you can pick up more information on the previous project from our website, and the link is shown there. We've actually been running since March 2011, and although it originally set out to be a 3-year project, we're now into a 9-month extension, and now very close to the end of the project itself. There's been, obviously, because this is a global pest, we've been dealing with partners in Canada, Japan, and the USA, and you'll see why Canada and USA and Japan, in fact, are important partners when I describe the pest problem. So it's important to understand the biology of pinewood nematode, because it doesn't act alone. It's actually carried around from tree to tree by an insect. And in this case, it's one genus of insect. It's a longhorn beetle in the genus Monocamus. And I'll put the cursor on to the name for you. The important point about the life cycle is that in its native range in North America, it rarely kills trees, but the beetle does need weakened trees to breed and to, in fact, carry the nematode from tree to tree. So the important part of the cycle is the transmission by oviposition, which we've got in the center of the diagram. And this is where the insect actually goes to weaken trees. The female lays its eggs just under the bark, and then the life cycle proceeds. The larval stages feed under the bark immediately, and then eventually enter the wood, burrowing quite deep into the wood. And towards the end of their life cycle, on the left of the diagram, they form a pupal chamber, and just as the insect pupates, the nematode actually migrates to that chamber, changes into a particular stage of its cycle, and then climbs back on to the insect. So the insects then emerge, the new adults, carrying the nematode, and they always feed in the crown of a tree on fresh foliage. And it's at that stage that the nematode potentially can enter the tree again. If the tree is resistant, as is the case in North America generally, nothing happens, the nematodes die. But if the tree is susceptible, then in fact it can die very rapidly. And this is what happens in the so-called pine wilt syndrome, which we'll talk about shortly. So the key facts then are that this nematode is native to North America. It rarely kills anything, so just to reiterate, it's more a saprophyte than a pathogen there. Lives on fungus in the tree rather than on the living tree itself. It's moved, though, in trade throughout the world, and generally this has been because wood has been traded either as a commodity in itself or as wood packaging, and that contains the vector insect, and these then fly out in the new country and can transmit the nematode into native trees. And this has happened in China, Japan, Korea, Portugal, and more recently in Spain, and also in Taiwan. So it's moved quite far around the world. And in these areas, it actually can kill trees very quickly indeed. And there's a photograph of the European vector, Monocamus galloprovincialis, and this is the one that we've been working on in Spain and Portugal. So pine wilt is the syndrome, as we call it. This is often called a pine wilt disease. Trees can be killed very rapidly indeed, within about 6 weeks. And basically, the nematode enters the tree reacts, often producing copious amounts of resin, and these tend to block the water-conducting system, the xylem itself. And of course, once the tree is unable to take water in, then it dies very quickly, and you get this reddening of the foliage within about 6 weeks of the nematode being transmitted. What we have found in our research and in practice in Portugal in particular is that European pine species, particularly Scots pine and maritime pine, are very susceptible indeed. So they die very quickly. But they don't die if the temperature is low enough, and I'll come back to that later. But the converse of that is that any monthly average temperature greater than 20 degrees, combined with low rainfall, will actually tend to lead to wilt expression in the trees. And that's an important characteristic. So in Europe then, the problem is the nematode is a new arrival. It was found for the first time in Portugal in 1999, but we now know that it probably had been there perhaps several years earlier than that. Very difficult to detect until the trees start dying. But in the surveys that were carried out soon after finding the nematode, it appeared to be restricted to the Setúbal area near to Lisbon. But intense surveys were carried out in trying to eradicate the nematode, and it was soon found that it had spread to other parts of Portugal, and indeed also to the territory of Madeira, which is the offshore part of Portugal. The nematode continues to spread mainly through the activity of the vector insect, but also helped on its way by human activity. And 4 small foci of infestation have been found in Spain. These have all been eradicated, or at least are under eradication. So we're trying to hold it at the border and stop it spreading to the rest of Europe. But because of the concerns about the nematode, all the European Union member states are carrying out surveys of their forests to check for the nematode. So we do the same in the UK, and in fact haven't found anything, luckily. But we also survey wood that's moved in trade, and interceptions of both the nematode and the vector insect have been made a number of times. Thankfully, in the UK, we are actually generally at low risk, partially due to climate, but particularly because, in fact, we don't actually have the Monocamus vector species here. It's not part of the UK fauna, and likewise Ireland is also fortunate not to have it. So we've been working on this, as I mentioned, for 3.5 years now. And we're trying to bring together our information, and very shortly online there will be a new item called the Pinewood Nematode Toolkit, or PTK for short. And this is a web-based synthesis of knowledge, bringing together our information as well as information from other people around the world who've been working on this particular nematode and its vectors. So we hope to launch this within the next month. And the interface example pages are shown here. These are from our web page that we're currently constructing. Tells you a little bit about the project. We'll be having this online very soon. And also the toolkit itself will be a simple clickable system using a series of modules. And each module, such as management, survey, modeling, etc., can be clicked through, and you get some baseline information on each topic, and then you can drill down for detailed information, both our own information and international information as well. So, for example, this is one of the pages that you reach by clicking through. This is the modeling module, and this happens to be what my team has been working on, trying to model which parts of Europe are likely to suffer from pine wilt. And so this will give the information, and people can then assess whether the area that they live in actually is at high risk of wilt expression. And I'll show you some more detail on that shortly. So to give you some information really on components of the system, we obviously look at the nematode, we look at the vector, we also look at the trees, and then we try and combine all those three together along with trade. So one example done by one of our partners in Germany at JKI is that they've been looking at Pinus sylvestris, which is Scots pine, a highly susceptible species, and carried out inoculation of those trees under laboratory conditions, but quite big trees, 2.5 meters tall, to try and mimic what might actually happen in the field. And trees were inoculated with nematodes and then looked at over a period of weeks and divided into pieces so we could actually track where the nematodes had got to in each tree and what the trees looked like as the infestation progressed. And a lot of results on this, but just to give you one example, the figure on the left is very soon after inoculation, and the nematodes still pretty close to the section of the tree where we actually inoculated them. So the cursor is shown just up there. So a couple of days post-infestation, nematodes move just a little bit through the tree. But 60-odd days later, from the color coding, the nematode is actually throughout the tree, top to bottom, into the branches, and also down into the roots. So it moves very quickly through a tree, and by this stage the tree is actually close to death and showing very high signs of wilting, including the reddening of the foliage that's characteristic. So these data we think will be similar, in fact, in large living trees, and there we got some information to support that as well. Okay, here we have some histopathology of what actually happens inside the pine tree when the nematode invades it. Two different tree species, Pinus halepensis, one of the European pines regarded as fairly resistant, and Scots pine down the bottom, which is known to be highly susceptible. And the top pictures show sections using scanning electron microscope on the left or conventional microscopy on the right. And you can see the resin ducts show quite clearly no sign of degradation, and in fact what you'd expect to see in a healthy tree. Tree in the bottom, to put it crudely, is a mess because the tissues have actually degraded. The nematode has invaded the living parenchyma cells and in fact invaded the resin ducts, the cambium, and eventually the xylem. And really this is the stage that the tree goes through when it's trying to defend itself in part, and also the breeding of the nematode literally blocks the vessels and stops the water conductance. So what do we do about the vector? Well, we've actually been working hard on understanding what attracts the vector insect to the host trees. And we've now developed through the project, and colleagues in Spain and UK have developed a synthetic attractant called Galloprotect 2D. And this is actually now commercially available, and what we now recommend for monitoring and potentially for control of the monocamus vectors. The vectors are attracted very strongly to the odors of this attractant, and we've also been developing the most efficient traps to capture them and retain them in the trap. And you've got two types here, the so-called multi-funnel trap on the left and a cross-vein, a cross-trap on the right. Both are equally efficient, but the key thing on both trap types actually is that they must be Teflon coated, in other words, made non-stick. So that in fact, as we often do, we wish to capture live beetles, and they're remarkably good at escaping from traps unless we have this Teflon coating. One of the reasons for getting live beetles actually is that we like to check to see if they are actually carrying the nematode, because obviously the beetles themselves can breed on all sorts of weakened trees and may not necessarily be carrying the nematode. So we've been looking at the potential for using these traps to so-called mass trap the monocamus species and reduce its density in the local area. Work in Spain by Juan Pajares in University of Valladolid has actually shown, at least in the Spanish conditions, that spacing the traps every 150 meters or so can remove up to 70% of the beetle population, around about 60 beetles per hectare. That sound a lot, but actually they can transmit an awful lot of nematodes, and they go from more than one tree when they do the transmission. So this could result in a lot of trees becoming infested. Work in France by a colleague there and in Portugal suggests maybe a different trapping density, but essentially also showing that the potential for trapping is rather good, and we are currently investigating the most efficient way of trying to reduce the vector population in this way. One of the other interesting questions we're always asked is, how far do these vector beetles fly? Well, we have a couple of interesting techniques we can use to try and demonstrate this. One of them, strangely enough, is to use what we call a flight mill, where we actually tether the beetles and then they're on this rotating arm, and as they fly, the arm keeps going round and round, and then we can measure the number of rotations in the flight mill, and from that we estimate the total flight distances. And although this is slightly artificial, actually it does match reasonably well what we see in the field. And you can see the data at the bottom. These are studies actually done in Austria on a different species. This time it's Monocamus sutor, which of course is potentially a vector if the nematode gets further north in Europe. And you can see the flight distances are roughly anything between 1.2 and 2 kilometers. And this is just in one continuous flight. So they can fly for quite a long time and literally just fly until they're exhausted. So of course, that's fairly artificial, of course, but we can then test that sort of assumption by using what we call mark recapture experiments, where essentially we take beetles we've found in the field, put a mark on them, some sort of indelible mark, and then release them again, and then recapture them using our lures and traps. And the distances they fly and the density relationships that we find in the mark recapture gives us a measure of dispersal distance and also population density in a given area. So this is quite a powerful technique for studying insect behavior. And for example, here's some data from a mark recapture work done again in Spain. And the distance, as you can see, for both immature beetles, these are beetles within about 10 days of emergence, and mature beetles having already fed in the foliage, they're very similar dispersal distances, anything up to 2 kilometers. And this is a fairly typical result. And of course, this is a series of flights within a very short time of the experiment starting. What we have to try and estimate is what is the lifetime flight distances. And we think this can exceed 20 kilometers. So they can fly quite considerable distances and hence disperse the nematode over those same distances. And we've obviously been concentrating on the known vector in Spain and Portugal, the one I mentioned, Monocamus galloprovincialis. But we've been studying these other European vectors, and our colleagues in Austria have been looking at Monocamus sartor and Monocamus sutor. And these are potential vector species. They're not present in Portugal, but if the nematode moves outside its current range, then we're fairly convinced that these species will then take on the role of vector. And these are just simply data of captures over a whole season, from the early spring when they first emerge through to the autumn. And this is a fairly typical pattern, either a single major peak or 2 or 3 peaks, depending on the emergence patterns of these beetles in the field. So another major question the European Union have asked us to investigate is, well, okay, the vector insect is known to be the primary route by which the nematode moves around, but can in fact the nematode move without the vector, so-called non-vector transmission? And we've been looking at this from the potential of pieces of infested wood that might actually come into contact either with other pieces of wood or indeed with living trees. We carried some of this work out, which originally was planned under our REPHRAME project, but to speed it up, we also worked in conjunction with CHEP, which is a European pallet logistics company, and they deal with literally millions of pallet movements every day throughout Europe and the world. And so there is a concern that the nematode that might be present in a pallet could transfer from one to another. And what actually we found, this can be read in more detail later, but essentially if the wood has a moisture content of greater than 20%, in other words, fairly wet, but still around the so-called fiber saturation point, then if the wood is wet, the nematode can move into it. It moves very rapidly between one piece of wood and another, the so-called recipient. But actually, if the moisture content is less than 20%, then in fact no transfer at all takes place. It's almost like a complete barrier to movement of the nematodes. And what we know with wood in pallets is actually they do dry out naturally, or sometimes they are kiln dried in advance. And therefore, after a few weeks, the wood moisture content is too low to act as a recipient for the nematodes. And in any event, if any nematodes are present in a piece of wood, they decline to zero in less than a year. And so pallets in service that actually are reused, which is what many of these companies do, actually have very low risk over a relatively short time period. We're also interested in wood chips. Wood that's been chipped but contains the nematode actually is a very good breeding site for the nematode, because there's a lot of fungal contamination in such a chip pile, and in fact the nematode can breed on that fungus. And so our colleagues in Germany have been looking at whether infested wood chips placed into the soil, either directly in contact with the roots or at some small distance from the roots, and the roots can be damaged or undamaged. We wanted to see if the nematode could actually move into those trees. And again, to cut short this long description, the nematodes can indeed move from wood chips into trees through the roots, and in fact cause infestation of those trees. So this is a new finding from the project, and actually quite a significant one. It does mean we have to be very careful about the fate of wood chips and what their control over their destination should be increased, particularly wood chips used for energy, for example. That's relatively easy to manage in the sense that they're all burnt at the end of the chain, but of course you need to be sure that they do go to a wood boiler rather than just dumped into the forest somewhere. So we're keeping an eye on this potential new transmission route. The other question we've been asked also is whether a tree that's infested can actually transmit the nematode directly to another adjacent tree through what we call root grafting or contact. And it's actually quite difficult to artificially graft trees together, and we haven't done too well on that one. But we have been looking at potential transmission from tree to tree in the same pot, which is what this experiment shown here attempted to do. And in fact, no transmission took place between donor and recipient trees. We've also done the same in larger trees in Portugal, looking at Pinus pinnata, which is the maritime pine host there. And again, after 1 year, although all the donor trees more or less had died, none of the recipient trees had actually died. And so it doesn't look as if tree-to-tree transmission through the roots takes place. So I mentioned earlier the modeling that we've been doing in my team. We've developed a process-based model looking at the photosynthesis of the tree and through its use of carbon for energy and defense. And we've been looking at temperature-dependent growth of the nematode and how the presence of the nematode can affect the ability of the tree to photosynthesize and use energy available to it. So we've been looking at current and future climates in Europe, and we've actually validated the outputs of the model against known impacts of the nematode in Japan. And we've also been looking at whether the trees die in the year of inoculation or in fact at a later time, maybe 1 or 2 years later, so-called latency. And the results are very interesting. We've now got a pretty good model that actually mimics very closely the real situation in different parts of the world. This is Japan. It's got a long known history. The nematode was first recorded as killing trees at the beginning of the 20th century, so it's been there a long time. It's killed almost all the pines in the southern part of Japan, and in fact has reached the northern end of the middle island of Honshu. And our model actually simulates very closely the known distribution of wilt in Japan, and that's accompanied by heavy tree mortality. And we're pretty confident that using these data to validate the model has given us a pretty good insight into how the trees are killed under this so-called pine wilt. So we've run the very same model then for Europe, and we have 3 scenarios here. This is a simplified version of the outputs. Red is where we think wilt is very likely, highly probable. The yellow circles are where wilt is possible, and this is where we have this so-called latency effect, where wilt doesn't take place the year of inoculation, but probably in the following year. And all the green bits, the safe bits so-called, are where we don't expect any wilt. And this has been run with, if you like, current climates using the summer of 2009 as just an examples data set. This was a moderately warm summer throughout most of Europe. Nothing exceptional about it, but very much reflecting the average. And you can see that the southern part of Europe has got a lot of red dots, and the continental parts of Europe, as you might expect, have got a very high likelihood of wilt expression. And the bits in Portugal where we've had mortality already fit pretty closely to where we've seen mortality. But we also ran the model for the 2010 data set. Now this was an exceptionally warm year, the hottest summer in 130 years, in some cases the hottest summer ever recorded. And you can see the expansion of the red dots, a great expansion of the yellow dots. And this is what we'd expect, if you like, under future climates, where you've got the extremes of temperature, higher average summer temperatures, lower rainfall in the summer. And this is a worrying picture because it shows that the nematode has the potential to kill trees much further north than its current range. But again, fortunately for us in the UK, the circles are still all green. And this reflects our maritime and much lower temperatures, maritime climate with lower temperatures. So one of the key things, of course, is to know how far this thing is likely to spread. And colleagues in France at INRA have been looking at developing a spread model, taking account not just of the disease expression, which I've just described with our models, but vector survival, how far the vector itself spreads, and then, of course, the accidental transportation by humans. And we're trying to integrate all of those into a probable model of spread for the future. And it's a complicated business, unfortunately. These are just some illustrative maps of the process we're going through. So we've been looking at human population density here on the left. So you've got hotspots of population density. Timber processing capacity is important because that reflects where wood is moved to from the forests, and therefore that's a risky pathway because the wood leaving the forest is at highest risk of carrying the nematode and the vector. And of course they move along mainly road networks, and so the maps on the right are the road network maps for Europe. So we look at a combination of these factors: human population size, wood processing, and therefore the forest locations, and how they moved along the road systems. And we come up with various risk profiles for those. I won't go into any detail on this, but essentially we've looked at, if you like, two extreme scenarios on this. And the one on the right is where most of the criteria are fulfilled. There's close proximity to forests, high density of population, and also very good road networks that carry wood from place to place. And in that sort of situation, we can expect the center of Europe to receive this nematode much more rapidly than, say, the southern parts of Europe or indeed the far north. So those are the elements that we've been studying mainly in our research. So we've been looking at the various elements of the science of our research, and then, of course, one of our major requirements is to disseminate the information. And many of us in the REPHRAME team actually are part of other international groupings that allow us to disseminate the information very readily. So the International Plant Protection Convention, IPPC, has a number of international standards and so on, and several of us have been involved in developing those standards. The European and Mediterranean Plant Protection Organization, or EPPO, has a forest quarantine panel, and several of us have been working on that panel on giving information on pinewood nematode, and so on. There's a number of other groups here that I mentioned in this slide. And one of our major outputs, in fact, which occurred a year ago now, was a large joint international conference between the REPHRAME project and the International Union of Forest Research Organizations, or IUFRO, Pinewood Nematode Group. We had a very successful conference in Braunschweig in Germany, organized by JKI, who's our beneficiary 5. And we had a decent number of people from 23 countries around the world. And in fact, if you're interested in reading more about the output of that conference, you can follow this link, and you can still download the proceedings from the conference. And in fact, this was just one of many dissemination events that we've held. And we recently had another workshop in Spain and Portugal for people to hear and see on the ground the work that we've carried out. So just to finish then, REPHRAME, in fact, as I mentioned already, is a European Union project, and it's part funded by the CEMF Framework Programme under this grant agreement. And this is the web link. Thank you very much.